So I think 2 hours is a long time to sit and listen to me. So what I'll do again is break it into 2 parts, just like I did this morning. Take a break halfway through. Does that seem reasonable? The other option is just to sit the whole. No, I will continue on. This afternoon, is the first of 2 lectures in neuroscience. And I'll apologize in advance that typically when I teach neuroscience, this is a 10-week long class. So what I'm going to do is just give you the highlights in just a few lectures. So I can't give you all of neuroscience, but I can give you some key parts of it. Those of you who have had neuroscience will know that I'm skipping over a fair amount of material, but I think so that we're all on the same level, I feel obligated to get us up to speed here, okay? So what I'm going to do is give you the recap of where we were this morning and why we even want to look at neural anatomy. I'm going to give you a bit of the overview of the vertebrate, our nervous system, okay? And then sort of the organization of the nervous system from the major pathways, spinal cord and central nervous system organization. And we'll do maybe one experiment with you today, which should work. It's an experiment that involves hearing a sound and interpreting it while you're watching somebody say that sound. And it's playing on 2 different circuits in your brain, and it's deliberately going to break the circuits, okay? So this should be fun, and it's towards the end of today's lecture. In the handout, you have a reference to Brain Tutor 3D, which I have on my iPhone, and you can download this. It's free, but I was just going to quickly show you that everything I'm going to talk about, I have to find my brain here, is an app that lets you look at the whole brain, rotate it around. You can get any region of the brain. So if you're not familiar with brain anatomy, this is a nice little app that you can download. I think it works on both iPhones and Androids. You can take sections through the brain. Let's see if I can do this here while we're talking. So I can take a human head and move through and take different sections and look at regions of the brain. So if you're not familiar with brain anatomy, this is a nice little app to give you a bit of orientation, okay? It's called Brain Tutor 3D. The other thing is the Brain Explorer, which is on the website of the Allen Institute of Brain Science, AIBS. And that's much more about cell identity. Those of you interested in drug delivery and neuropharmacology will want to understand that the brain cells themselves are highly regionalized and biochemically highly specialized, okay? So this will help there. And a great paper here that I think is worth reading. And I will probably upload the PDF, or at least the link, although you can type that link in, okay? Great. Okay, so this is where we kind of left things, that we have a very complicated multipartite structure. And when we teach neuroscience, there's really 2 philosophies that we can use. I can say I'm going to start with the molecules and build it up to something large and understand the organization of the brain, or any brain for that matter. Or I can start at the large scale and work my way down. And given that this is dominantly engineers, I've decided to start at the large architectural scale and work towards the molecular, okay? You can go either way, it doesn't matter. The challenge we had is this reverse engineering problem. That is, how do you understand brain functions, signaling, interpret large signal sets? Part of what we need to do eventually is understand when we're recording from a brain, whether it's human or Drosophila, the fruit fly, or a moth or a frog or a mouse, it's the same problem. It's how do you pull out data from a rich and complex set of signals, okay? That's a big open problem. And it's been solved in some ways, but it still remains lots of room for improvement. Okay? So let me kind of give you the overall architecture of the brain, and bear with me as we do this. We'll come back to this shortly. Typically, you divide the brain into the central nervous system and the peripheral nervous system. And centrally, it makes sense, it's the brain and spinal column, okay? There is a great deal of information to focus on here. I'm just going to quickly go over the peripheral nervous system. So as it suggests, the peripheral nervous system is the part of your nervous system that feeds into central. So it's all the sensory neurons coming in, all of the motor neurons going out. In neuroscience, they'll refer to these as efferent pathways, things leaving, and afferent pathways, things coming in. Everything coming in is sensory from the periphery. So auditory information is coming in peripherally, tactile information, temperature, pain, everything comes in peripherally, is synthesized initially in the spinal cord, processed there to some extent, as you'll see, and then also sent up centrally for further processing. Okay? We'll talk about timing and delays and sort of the circuitry of these in a little bit. So you have a sensory division and a motor division. You have internal and external sensing. Okay? So some biologists call this, and it's a terrible word, but proprio or internal reception, and exteroreception. So again, anything about environmental signals coming in is external. Internal states, carbon dioxide levels in my blood, state of stress of my muscles, mobility of my intestines. These are all sensed. Some of it you're aware of. Most of it you're not. Okay? Under the motor division, we have these two categories, both of which have been subjects of considerable attention in neuroengineering. There's the autonomic nervous system, which is oftentimes confused with the word automatic, but it almost means the same thing, self-controlling part of the nervous system, and somatic, voluntary. So for example, how many of you had lunch? And if you didn't, you should. It's an important meal. Your stomach is undergoing incredible motions to move things through, to churn, to move the intestines. Things are moving around. That is all autonomically controlled, right? Most of your respiratory circuit is autonomically controlled. You cannot consciously control your intestinal mobility most of the time. Typically, you cannot consciously control your heart rate. Although, with appropriate feedback, you can learn to. But the baseline is that it's just autonomic, okay? The somatic system is anything you can do voluntarily. You can speak, you can reach, you can strike one another. I'd rather you don't. But these are things you can do, okay? Let me just look a little bit more at these two autonomic nervous systems. So that arrow should be right here. In the autonomic nervous system, it's actually two competing pathways that are always operating at the same time. And in fact, these have been subject of some neural engineering interfaces. So for example, you have the sympathetic nervous system, which is also called the fight or flight. And that's like when you're scared, when something frightens you, right? You launch an entire series of neurally controlled processes. And these include an increase in your heart rate, right? Your airways open up because you're trying to get, you're under a stress situation. You need more oxygen. You're breathing rapidly, right? All of those things are happening in response to fright. If I were to come up and threaten one of you right now, I won't. Your heart rate will go up. Your mouth will get quite dry, actually. Anytime you're nervous, your intestines will be inhibited, and the world continues on. In contrast, the parasympathetic system is a slowed heart rate, salivation, constricting airways. Your stomach is going, it's this sort of rest and relax, and it's the part of your neural system that's undergoing contrast to that activated one. The thing is, it's not like you're doing one or the other. Both are operating simultaneously, and it's just always a balance of these two. I mean, the particular case is the vagus nerve, and vagus nerve comes down and controls heart rate, and it has both pathways present in it. So the vagus nerve is both pushing towards greater muscle activity in the heart from the sympathetic pathway, and lower activity from the parasympathetic pathway. Both units are going on simultaneously, and you just have to learn to activate one or the other. One of the challenging things is getting a vagal nerve cuff for control of heart rate. And it's not controlling each beat like a pacemaker. It's actually stimulating preferentially parasympathetic or sympathetic pathways so that you can stay within control of if you have an aberrant heart rate. Okay? Candidly, something like a pacemaker is also, at the end of the day, like a neural engineered device. It's providing electrical stimulation to electrically active tissue, which happens to be heart. Okay. Parasympathetic and sympathetic nervous systems come from central pathways down the spinal column, right? And if you look at a cross-section through your spinal column, it's actually very organized. So pathways maintain a spatial organization right up through the spinal column. And you can use that organization for very clever interfacing with electronics. And I'll give a couple examples. This is a cross-section through the spinal column. This is your back, and this is your stomach, okay? So this is the dorsal, or back, and ventral pathways. The dorsal root ganglion is all sensory. So this is really interesting. Right here, the nerves coming from your body come in regionally, right? And they split like this. This pathway is all sensory information coming in. This pathway is motor information going out. So if you wanted to control motor activation in your spinal cord, you want to stimulate only motor, you wouldn't go in here where they're mixed up. You wouldn't even go in here where there are neurons that are connecting the two. You'd want to come right here. And it's actually just coming out of the spinal column that's past the vertebrae, so it's accessible. If you wanted to block sensory information from your body coming in, you would probably want to go right here. Not in here, it's too hard. And as you'll see, you can do this very, very regionally, okay? So let me just go back here. This spinal cord is broken into 12 thoracic, 5 lumbar, and 8 cervical units. There's neck, thorax, lumbar units, and each one has its own circuit, all right? So this is still in here, inside here is still central nervous system. Right up to here, this is peripheral now, and this is peripheral. It comes in and makes a connection in there, okay? It's regionally organized. And these are developed somatically. And by the way, if you look at this map, and the map I showed you this morning of acupuncture sites, these are very, very similar maps, okay? So acupuncture, as we said, was discovered over hundreds of years of observation, right? This is a more recent sort of technical stimulation mapping. Just to be very clear, one side, this is the front and the back. These are called the dermatomes. These are sensory maps of your body. Every bit of sensory information is coming up here, and it's processed right in that part of the spinal column. It's not processed down here or over there. That's its initial processing, okay? So if I can feel something here, right, I can tell you when I'm rubbing this exactly where in my spinal column that information is being sent, right? And if it's here, I can tell you exactly where. I just have an interesting side observation that I always tell people. There's a, if you had pox, smallpox, or if you had herpes pox or any of these, there's a virus called the Herpes Zoster virus. And it's a virus that actually lives in the dorsal root ganglion. That's where it resides. And it lives there for a very, very, very long time. So if you've been, chickenpox is the one I meant, chickenpox. If you've had chickenpox, that virus stays with you almost your whole life. Most of the time, you're keeping it at bay with your immune system, okay? It doesn't sound like this is at all relevant to the class, except it's a virus that resides in the dorsal root ganglion, that's the sensory input world of your body. When you are under great stress, just as an individual, your immune system is compromised. You tend to get sick more easily. And as it turns out, when your immune system is pushed down a bit, that herpes zoster can actually travel. And it travels, interestingly, this way down your sensory nerve. So even though electrical information comes in one direction, the virus travels all the way to the nerve ending. And it's called shingles. And when you get that virus, you can see exactly where all the nerve endings are coming out because it bursts out of the nerve endings into your skin that those nerves go to. So this is one dermatome exactly mapped out by that virus. And so you can exactly tell, if I go back a few slides, that dermatome was right here, right? That was C6, T6. If you look, T6 came around like that and wrapped in. Now let's look at the individual so I can tell you exactly where that was. And there it is, wrapping around, coming up slightly around the back, just like we expect it to. That's where that virus was residing. All that is a demonstration of the precision of the spatial mapping of nerves into your body. Extremely precise, okay? Great. So we have a dorsal root ganglion for sensory information, motor information coming out. This is sectionally organized into the, let's make sure we're still dorsal. There's processing goes on in here and here. So probably the best thing I can do is do a demonstration with one of you. See, is there something? Yes. So I'd like a volunteer. Good. Great. Come on up here. Please join me. I'm going to demonstrate something you all know exists. Please have a seat. Relax. Make sure your feet are hanging, not on the ground. That's great. You sit straight up, and that's great. So what we're going to do is give him a strong sensory stimulus, okay? That's going to come in through his dorsal root ganglion, okay? That strong sensory stimulus has to be processed extremely quickly for him to prevent injury, okay? And we're going to rely on a circuit that resides right in here in the lateral gray horn of his spinal column, okay? And as it turns out, okay, can I have you sit a little straighter up? Are your legs hanging nicely? Yeah, they are. So on the left side, if I hit, there's a reflex. It's not very good. Sorry. There we go. So that went very quickly. It's a reflex arc, and it was processed principally by his spinal column, but he was well aware of it. So I'm going to ask him to do another experiment. So I want you to close your eyes, okay? So I don't want you to see this coming. I want you to, before you do, okay, open your eyes, okay. Take your arms and pull hard like this and close your eyes, okay? So while you're doing that, I want you to stop your leg from moving the minute you feel the impulse, okay? So if his brain is super fast, it's not going to be a problem. Nope. Notice I didn't tell him which leg. Okay, both legs, are they relaxed? I'm just checking. Can't do it. The leg is all the way out by the time he feels it. Well, why? So, thank you. You can go. Unless you want to stay here. Oh, no. Let's do a little examination. There's two kinds of reflex. This is a reflex arc, and that was done in the spine. So the main goes like this. So this is not the particular case in his, but there are different ones. But let's take one example, which is if you put your hand on a pin, and that's not an experiment I'm going to ask you to do, but you can imagine you pull it up very abruptly, correct? That sensory information is brought in through the dorsal root ganglion, and this is an interneuron. So there's sensory neurons and motor neurons, and they're interneurons. We'll talk a little more about how they do the processing. This is the most elemental computation possible. If sufficient information comes in to this interneuron to get it to command, to make a command, and I have to talk about that tomorrow, that command is launched in two directions. It is launched first to the muscles to cause that, and secondarily, it also sends a pathway up the other side, right, to the other side of your brain, okay, because you're crossover right there, so that he is aware that something happened. So you did feel it, right? Good. But there was no evidence by the leg motion that he felt it, because he couldn't stop the leg from moving. It had already gone all the way, right? And that's not, I mean, that's normal. So that's good. So that has to be processed. This dynamic right here, right, takes time. And that's because it's not so much that the signal travels very slowly. It's that the step of the signal transmission from one neuron to the next has a fairly significant time lag, okay? It varies from connection to connection, but right away I had to do 1, 2 connections just to do this. Now let's think through what would happen if I had to do this by this structure up here. So everything, leave this off, goes all the way up, comes back down this side, crosses over, and then initiates it here. So I'm so curious, why would this take longer? I see a yes. Why?
Because the path is long.
So it's a longer distance. Any other reason? More connections. So why are there more connections?
In your brain, you may first the signal first travel to maybe thalamus, and it pass to your cortex.
Right. So here's what happens is, as you'll learn, is coming up my spinal column, there are very, very few connections until it gets to the base of the brain, the thalamus. That signal has to come up, and it goes actually in several ways. First of all, it goes into the sensory cortex, as you'll see. I have to know that something happened. And then, if I have to do something about it, I have to plan the motion. That's called the premotor cortex. And not only do I have to plan the motion, I have to launch the execution. That's called the motor cortex. And then I have to send all those signals back down. So there's probably 4 layers of connections that I have to do. Each one of those are possibly multiple connections, obviously, and each one has delays. So it's not just the duration, the distance, but it is very much the number of connections. It's both, okay? If I were guessing which one's more important, does anybody have an estimate? Is it the distance that's going to be more important or the number of connections? I think you're all correct. Why do you think that?
The signal transfer from one neuron cell is so fast, but between 2 cells is slow.
Right. So signal transmission is quite rapid, but not infinitely fast. It's still has a finite time. As we'll talk about later, we have specialized mechanisms that permit rapid signal transmission in our neurons. In certain disease states, multiple sclerosis is the biggest one, or there's another one, modify that. It's called Guillain-Barré syndrome. Okay. Very good. In the case of a muscle spindle stretch, which is a little different again, that's actually a monosynaptic connection that's hardwired. That one, the only reason you have central awareness of it is there's a parallel pathway that this axon makes to another interneuron set that goes centrally. But a lot of the state of, as I'm walking, okay, let's take that example. Much of the processing for walking is done right here in the base of my spine. Most of it. You can get a human with spinal cord lesion, so they can't walk. You can put them on a treadmill, right? And you can get one leg, as long as it's weight bearing, right? You get one leg moving, and it induces the other leg to do the contralateral activity. So the circuit works down here, and a lot of it does. In the US, there's an expression called running around like a chicken with its head cut off. Okay, does anybody know what I'm talking about? Oh, I know you do. So it is, if you cut the head off a chicken, it runs just fine. That whole spinal circuitry is a fantastic system. So again, just a point of reference, one of the issues in neuroengineering is we've had a tendency to want to control appendages by muscle. That's been our thinking, kind of like how we would do a robot. What the new thinking is actually, could I control the pattern generator that resides in the spinal column? So that is, it's already got all the programming built in. I just need to launch that device, right? So this is why I'm bringing this up, is the spinal cord is already central nervous system. It's already doing computing. It's already generating pattern. And it's a very primitive part of our brain, okay? Excellent. So let's go up north into the brain, okay? And let me give you a sort of a landscape. I'm just going to do a few examples. So this is really fun. I'm not going to teach all of brain science in 20 minutes, but I'll do as much as is reasonable, right? So the brain is very, very regionalized. And it has, let me give you a sense of the anatomy. People tend to think of these as very random, how they're built. That is, the brain is just a sort of a bowl of junk. Actually, these are very, very similar from individual to individual. These are each called the gyri, or gyrus, okay? And the mapping of the various gyrus is on BrainTutor 3D. It's a free app. I'm not advertising it. It's just, you can get it, okay? Between the gyri are things called the sulci, or sulcus, okay? Those are the valleys. And to give you an idea of how we tend to think of it, can I have a blank sheet of paper from someone? Thank you. I'm going to ruin it. Okay, so the brain, as you'll see, the cortex in particular, is structured into layers, okay? Right now I've just got 2 layers, very finely mapped up. So the way to think about your brain is not as a volume, but as a huge sheet that has to fit into a specific volume. And as you'll see, these layers are actually structured in a way that there's communicating lines within a layer, okay? That's intra-layer communication, and there's communicating lines between layers. And that architecture is the architecture that motivated neural network the concept of neural network architecture, okay? So there's connectivity this way and connectivity this way. And in fact, as you'll see, there are many layers in the cortex. The problem is that this sheet-like organization has to be filled up, squeezed into a smaller volume called your brain case. And the way you do that is you wrinkle up this sheet of paper into this, and now I have sulci and gyri, gyrus and sulcus. And those are these folded, highly folded layers of the brain. But conceptually, you stretch it out, and it's all mathematically and computationally like a giant multi-layer sheet. Okay? So that's your first order of business. Let me just see what my next slide is. Okay. So let me just get you oriented. This is the front. This is the back. This is the most primitive section of the brain, the cerebellum. And it goes into a region called the medulla oblongata. So this region here is pretty much like a super advanced part of the spinal column. But it has pattern generators, your respiratory rhythm. A lot of the basic functional rhythms of the brain are carried out right there. If that part is destroyed, you really, that's not fixable, okay? Then you would be on, even in this case, you would be on a respirator, and it'd be a very, very difficult situation. So it's not repairable, that we know of. This is slightly more advanced, so this is post-motor programming, but there's still motor activity, cerebellar control of movement and dynamics. So the patterns that are launched, pass through here, are conditioned into muscle outputs, okay? And then this region here is hugely associated with sensory information, as you will see. And then this is the planning region here, okay? So let's break this down a little more. And so if you're going to put electrodes in each other, know where these go. So this is planning, speech making, decision. This is not just speech making, this is speech recognition. This is also areas where you recognize written words, heard words, where you plan spoken words and you plan written words. There have been fMRI studies done of different individuals from different cultures, and there is evidence that the region of the brain for written speech in Asian cultures is slightly more expanded because you have apparently a larger character set than we do. Just an interesting datum. I don't know where I got that, but it's fairly recent. fMRI, functional magnetic resonance imaging. Visual processing is here. Visual information and auditory information come in and are processed in here for your ability to follow a talk, to speak multiple languages. I was talking to Yiming earlier that as you learn different languages, they are actually, those languages are parked in different parts of your brain. And the reason we know that is if, as you get older, and you may or may not get a stroke as you age, much of what we learned about the brain, as you'll see, comes from individuals that had lesions or a stroke or a region of the brain destroyed, and it's the loss that happens after that region is destroyed. So somebody who had ancestrally learned, like my father, for example, is an interesting example. He was German. He grew up speaking German and then moved to the United States, learned English. He moved in the late 30s, early 40s, and learned English. And then, when he was about 80-something, he had a stroke in that part of his brain, and he lost his ability to say certain words in English, but could say them in German. And the fMRI studies show exactly where those English words were stored and processed. It's just a fascinating regionalization. Most of you in here are bilingual, because I've heard all of you now speak, and you're all great. Your English section is parked differently than your either Cantonese or Mandarin or whatever your particular language is, okay? And your brain spends a lot of time not just acquiring information, but wisely storing it. This is something we haven't done in synthetic systems, is to be able to easily move our storage things around. So that as I acquire a new language, I move sites around and I take things over, right? Really fascinating. Here are examples of pathways this is showing here. Again, this is hearing and advanced visual processing. That's your temporal lobe. This is all regions of your cortex. I just wanted to make a little clear here. This is the prefrontal cortex. This is motor. This is somatoesthetic or sensing, okay? So now we can do a little more regionalization. Turns out this is extremely, extremely robust among individuals, this separation here. So right in this band here, right, is the region of your brain for planning the execution of tasks. Remember I asked all of you to imagine grabbing something and squeezing your hand? That was lighting up, and I can tell you exactly what part of the brain that would be happening. If you're squeezing your right hand, right, just like this, and you just go right above your ear canal, straight up, your right hand is mapped right to about that point for the premotor cortex, maybe back a little bit. Then it's the motor, and then it's the sensory. So I'm feeling this here, I'm squeezing with motor activity here, and I'm planning it right here. And you literally, you can map that. And that tells you that if you need to place electrodes in a brain for assessing certain activities of certain parts of your body, it's extremely reliable. And it's well mapped. That's the most important thing you need to know, okay? Keep grabbing this chunk of brain here. Here is the mapping more precisely, okay? And on the left is sensory, and on the right is motor, okay? So again, there's motor and sensory. Let me just go back. So sensory, motor, premotor cortex. This is frontal, okay? So now, here again is the somatosensory and motor. Notice they're not very different. That is, the amount of brain volume you devote to a particular function is not very different than the amount of, to sensing it, is not very different than you would devote to controlling it. For obvious reasons, your hands, right, occupy a very, very large fraction of your cortex. We are very, very, very focused on our hands as our primary tactile sensory organ, and it has an amazing amount of manipulation. So as I was telling you earlier today, that this problem of manipulating a cup with fluid in it so I don't spill it, right? That's a hard robotics problem, but we've generated and devoted a huge amount of circuitry to this, both in the sensing and in the control. And that's an important lesson for those among you who are roboticists. That is, if you just focus on the control and not sensing, you're missing half the picture, right? Your face and lips and mouth. So if you think of humans, what we are grabby, speaky things, right? As opposed to other animals. I could give an entire lecture on just sensory amazing things about things that aren't human. My favorite is something called the star-nosed mole, whose nose, just the nose, occupies in its brain the same as our hand. And they use their nose, which has fingers all around it, it's called the star-nosed mole, to feel vibrations of the earth where earthworms are moving, and that's what they eat. And they devote a huge amount of their brain to it. It's their hand, right? I think there's a couple interesting things here. From the sensory side, look at where your thumb is, right here. This is your hand, and here's your face. They're quite close to each other in the mapping of your brain. Okay? Oh, that's the, if you were to make a clay version of a human, It's a mouth with hands. It's sort of what I feel like in front of you, just this mouth. And, okay. Why did I bring up this hand? Many times humans will have amputation. Cancer, loss, injury, or whatever. Turns out that if you lose this part of your arm, right, your hand maps into your cheek. And you can actually rub a person with a fairly recent amputation here, and they begin to get phantom feeling of what's out here. It doesn't exist anymore. That hand is gone. But you can initiate a feeling of touching something by going into regions of your brain that are beginning to connect to where the hand was. Let me just go back and show you another picture of this. Sorry about the back and forth, but what's happening is right in here is your cheek, right? And it's a little gory detail about the mapping here, but these neurons actually are really, really close to each other right in here, okay? Particularly the upper cheek. And remember, it's this sheet that's connected. Those connections are just moving in this direction strongly. I find that fascinating. Again, it's very plastic. It changes. If you lose an appendage, you rewire. Just like the monkey was able to grow this new arm that we talked about earlier. I'm going to do a couple more examples of circuitry here. This is how the visual world works. And just so you understand that when people are beginning to work on visual prostheses, that is, I'm going to help a blind person see. Okay? Many kinds of blindness. One is loss of retinal pigments. That's a big one. One could be neural loss, loss of the optic nerve. Some individuals get cancer of the optic nerve, right? There are a lot of ways in which vision may be lost. So the trick is, if you were to step in and want to stimulate regions for vision, where would you go and how would you do this? I'm going to give a little more detail on the circuitry of vision. So bear with me, okay? So you process visual information in the back here in your optic lobes, which is the visual cortex. This is probably the most studied part of the human brain. It's called V1, the primary visual cortex. It is the subject of study now at the Allen Institute of Brain Science. And the reason is it's where the most primary visual computations are done. So once images are coming in by the optic nerves, how does your brain make sense of what images are and where do you begin? Okay? So that process is done here, but the information actually propagates from a region in the brain here. So now let's watch what happens. This is the optic crossover, or it's called the optic chiasm, and that's right here, okay? And here are these pathways that are coming around to the optic lobe. I mean, this is a massive part of our brain. We are extremely visual creatures. We really, really devote about 50% of our brain to sensing, and a good part of that is vision. Okay? We're not the most visual creatures, but very visual. Okay? So let me first tell you that things on the left side of the world and the right side of the world map in a complicated way. Okay? So watch this. This is the left side of the world, solid, and the right side of the world here. Each eye sees left and right, right? The left eye has left world that stays on the left side of your brain, right? Got it? So it comes in, stays left. But the left eye also has right side of the world that moves to the right side of the brain, okay? The right eye has right side of the world that moves to the right side of the brain and left side of the world that moves to the left side of the brain, okay? This side goes like that. This side goes like that, okay? These are odd. I can't help it. These are processed right in here and transformed in a map that literally takes the world like this, okay? So there's left and right, and it rotates it, crosses it over, and it rotates it so that it maps spatially in that region. So it's actually called a retinotopic map. That is, literally, I can get sectors, follow them all the way over, and I know that they map into the brain in a spatial array of the same orientation. Okay? So that's, again, more powerful information. If you need to stimulate the brain to see things, you completely know that it's got a map that you can stimulate and give dot patterns that can be perceived. And this has been done for blind individuals. You can directly stimulate the sensory cortex and know that if I stimulate here versus here, I'll get two different percepts of movement or light or something, or perception of it. Cool. Last story, and we're going to take a break. So I was at Caltech, and at the time there was this guy there who had just gotten the Nobel Prize. His name was Roger Sperry. And he got the Nobel Prize for figuring out that the brain has a huge left-right asymmetry in terms of how it processes information. You know, some people said the left side is artistic, the right side is analytic, or whichever, I can't remember. Okay? He ran into and recruited a series of individuals that had a lesion separating the two halves of their brain. Some of these individuals got the lesion by injury, right? Others, it was deliberately done. They cut the connections between the left and right side of the brain, and those connections are this massive connection right here called the corpus callosum. It connects all of your left world to all of your right world. And the reason surgeons went in and cut it, just completely separated the halves, is that that was a site of seizure disorder, a very common site. And the seizures are these uncontrolled left, right, convulsive depolarizations of your brain. Huge, giant left, right signals. And all they did is go, stop that, cut it, and you have your brain separated. So an individual with that separation in this area, the corpus callosum, the big connector, still has these crossing over, but they're missing some of the connections that connect the logic of the image, right? So there's your ability to see things, and there's your ability to process them, okay? And they took this individual and they had them look at a word, in this case, cowboy. It's a word that consists of two words, cow and boy. And the person who sees it, who's fully intact, could say, cowboy. It's, you know, whatever. But they projected it in a way that boy went on one eye, cow went on the other. So they couldn't see more than, they had to put it together in their head, right? When that happened, they say boy, they say the word boy and write the word cow with their right hand, okay? Left hand, sorry about that. I thought that would strike you as interesting. And these are all done, these connections are done through the optic chiasm, and this is called the lateral nucleus, and that's where that connection forms and is processed laterally, okay? Just one more personal story. I was on a flight in December from Seattle to somewhere. It didn't really matter where. And it's great being a neuroscientist because when things go wrong, you completely can analyze it. So I'm on an airplane writing a talk, which I tend to do. And I thought that there's something wrong initially with my computer screen because I couldn't see the left part of the display. Right? So something wrong. And then I thought, no, it's my glasses. No, something I couldn't see the left side. And I closed one eye. And I thought, oh, I can't see the left side of my computer screen. I thought maybe I have some, it's called retinal detachment. That's not uncommon, right? It's not great, but it's not uncommon. And then I said, oh, but I know neuroscience. I'll just close my other eye. And sure enough, I couldn't see the left side with the other eye. So if I can't see the left side, somebody want to tell me, is it in the retina? What's the likelihood of both eyes detaching in exactly the same? 0. Here? 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 Here or here? I can't see the left side of the world. This one? You're right. It's the lateral geniculate nucleus, the right one for my left side. And what it was a small ischemic attack. It's nothing bad, but it was interesting. And then you can sit there mapping it, and the person sitting next to you in the airplane is a little concerned, but like, what am I doing here? Just fun. If you know neuroanatomy, you can know a lot. There's an individual who had a severe lesion in the lateral geniculate nucleus. So control individual is given this picture to draw, right? And they can do that. All of you can do that. A patient, absent the lateral geniculate nucleus, is given this picture to draw, and this is actually what the patient drew. And that same person, if you give them a plate of food, right, they will eat everything on the right side of the plate, no problem, and then they're done. Food's gone because there's nothing on the left side of the plate. If you rotate the plate, they're fine. They can eat the rest of the food. So there's another interesting thing about the amazing processing done of the brain. Okay, I think this point it's good to take a break for, say, 10 minutes, resume at 3:05, and I just have a little bit more to do with you. And an experiment for the whole group. Great. I was trying to think of ways to look at very high-level brain function and information processing. And even though I think most of my research interests are in sensing and motor action, particularly with mobility, a fairly easy analysis is of language. And so I was going to spend a little bit about the understanding and acquisition and production of language. That is, speaking a heard or read word, right? Or interpreting them, okay? So I need to kind of get you oriented again into how the brain is going to do this. And it's all about information pathways, okay? So bear with me as we think about the problem of speaking a heard word. So if I say, Ngoy. Was that close? Was that close? Okay. Ngoy. Now you say it back. What? All of you. Say. Okay. Thanks. So you're hearing the word. It's processed by your cochlea, okay? That processing has to convert really complicated signal into something that you understand, okay? And that understanding is done principally through Wernicke center. So it's primary auditory, Wernicke, and then for you to form the word, you have to think about what the word is and the motor program that goes with it. So when I say it, you hear it, you process it, to form it, to repeat it, you have to then launch it into the primary motor cortex. And that primary motor cortex, if I ask you to say it, has to control your lips, your tongue, your throat, your larynx. It's amazing amount of programming that has to go on. So if I say the word and I ask you to repeat it, it happens like that. That is a really, really hard task, okay? Now, from a machine standpoint, if you wanted to synthesize something, it's called a tape recorder or a recorder. Simply absorb it, and play it back. But there's zero interpretation, right? None. It's just the sound is repeated. In this case, the sound is first heard, parsed, and actually it's a really interesting Fourier transform that happens on the sound, repackaged into a frequency-mapped region of the brain, then sent back into an interpretation on the sets of information that came in to what that sound meant, and then how you interpret it. So let's work our way through this, okay? And bear with me because it's kind of fun. So let me give you, I'll get to the experiment in a minute. So brain lesions, again, are where most of this information came from. There's a wonderful book that I'll make note of. It's by a very famous neuroscientist, Oliver Sacks. And he wrote a book called The Man Who Mistook His Wife for a Hat. And it literally was about this man who kept thinking his wife was a hat, okay? He had this disconnect between what he sees and what that meaning was, okay? But it dealt with a lot of these, what we call brain lesions, and how we have come to understand how brain function works by people who had those parts broken. So, for example, a break in something called Broca's center, I'll just go back there, okay? Or Wernicke's center. This says, you know the word, but you can't say it. Here's an example. An individual with Broca's center lesion, if you show them this, they know what it is. Can you tell me what this is? What is this? Yeah, they can't say that. They say, it opens a lock. They can't say the thing that they see, or they have difficulty in doing that. In a lesion in Wernicke, Wernicke is the association of what the sound or the image is to its actual content. So I have no idea what this is. I can talk about it. It's shaped like this. I don't know. I don't know. I just don't know what this is. And that's that lesion in the association cortex, okay? Again, I'm emphasizing the regionalization of the brain, okay? So let's pick up a few of these. This is Broca's aphasia. This is a severe loss of Broca's function, okay? It's Brodmann area 44 and 45, and Brodmann areas are these principal regions of the brain, okay? And you can look these up. I'm, again, brain tutor, if you wish, right? Non-fluent, labored, hesitant speech. It's not like it's gone. I didn't want to exaggerate, but most also lost the ability to name persons or subjects. So I can't name things either. I can utter automatic speech. I can say hello. Comprehension is relatively intact, although it's difficult to assess because the person has a hard time communicating with you, right? And it really depends on the extent of the stroke or lesion. If it's very extensive, they're not speaking at all. Most have some partial paralysis on that side of the body. So that's because the region is right up against that motor cortex. So if you lost that, chances are you lost some motor area as well. Not much recovery over time. Doesn't remodel, okay? In Wernicke's area, and I'll go over this later, this is lesions in the posterior left temporal gyrus, right? So there's Wernicke, there's Broca, right? Right here, right? You can speak, but you contain many weird things. Like you may confuse girl with curl. It's very easily done. They're conceptually quite different. You may confuse bread with cake, right? So even though girl and curl sound alike, they mean something very different, but bread and cake are both baked things, and they kind of lock up together and get confused easily. You have oftentimes syntactical but empty sentences, meaning you're just saying things, but they mean what? Chair, moon, hello. Chair, moon, hello. That doesn't mean a lot, but they tend to lapse into this. You can't repeat words or sentences easily, right? You're unable to understand what you read. So it's very difficult to process that information. Now, again, these are examples. They're not all uniform, and some of them have various levels of intensity. So different individuals will present with different levels of that extensive loss. So some are quite functional and just little bits are lost. Some are simply unable to communicate, okay? So let's watch the process, okay? Yeah?
Like, when do these aphasias arise? Is it generally after strokes?
Stroke is the dominant stroke, and there are many kinds of strokes, but usually it's what we call lesions of the brain, a loss of a brain. So to be very clear, a stroke can be an aneurysm. That is, you have blood loss and therefore circulation loss of that part. Those are very bad. Those are very extensive. Or they can be a blood clot restricting flow to a critical part of the brain. And even if it's brief, meaning hours, that's a very long time for neurons to live without oxygen. So those get lost. And even if the clot isn't there, that part of the brain tends to not recover well. Although, again, these what we call transient, coagulative, clot-related strokes, those, if they're very brief, you get good recovery. Okay? Did that answer your question?
Like, does it happen through traumatic brain injury? Or?
Yes, TBI. Yes, absolutely. Traumatic brain injury, this is something we should get to. This is associated with car accidents is a big one. Sports is very big. In the United Football is a big source. Hockey. Somebody's Canadian here. Sorry. The more common presentation is cognitive loss, because it's a lot of frontal lobe cognitive stuff that we see the evidence of more readily. And again, traumatic brain injury tends to be a little more focal than stroke, okay? It's great questions. Excellent. But TBI is actually a big source of some forms of paralysis, although motor loss from traumatic brain injury is not that common. It exists, but it happens. Great. So let me work you through this, okay? Just a fun example. So let's follow repeating a spoken word, the example we just brought up, okay? So your sound is analyzed, your primary auditory cortex. So we find this right here. So sound is acquired, obviously, by your ear. That sound is transduced by a structure in your ear which separates the sound. So bear with me. Your ear has inside it the cochlea, right? Is that familiar? It's a coil of sensory neurons. If you uncoil it, right, the ones closest to the opening of your ear processing high frequencies, and your sounds are frequency separated. So it's basically an auditory Fourier transform of the sound. That Fourier transform, right, is measured by neurons along the length, okay? Low frequency near the auditory opening, higher frequencies towards the tip of the cochlea. That map is actually brought into the auditory cortex, and it's frequency mapped on the auditory cortex. Low frequency at one end, high frequency at the other. So that's called a tonotopic map. Remember I said there was a retinotopic map, the sides of your retina, map on regionally. This tonotopic map, or frequency map, is mapped onto your brain in that order. Just like my face is mapped like this, it's mapped literally, physically onto the cortex. Okay? Cool? There are lots and lots of these maps we can talk about, but anyhow, that's the acquired. So this happens here. This information then is passed to determine in this part of the brain, this is Wernicke. Remember, that's your conception of what it is. And so it says it analyzes the sound to determine what's said. What I find fundamentally amazing is how fast this is happening in you. Right now, I'm saying words that I'm talking pretty fast. There aren't many computers that can do this. And by the way, it's a really hard problem for my voice to understand, for a computer to just get one voice down. Then I hear maybe your voice or your voice. They're very different tonal structures, very different frequency spectra. They're processed, and we completely know what we're each saying. There may be some challenges and differences, if it's soft or quiet, accent strong or weak, right? We can still do this. That's just amazing, and we're doing it lightning speed, right? And that's what this structure is designed to do. Birds have to do this too. I just wanted to point this out, that one of the model systems for learning of sound and interpretation is in fact the bird brain for song. And this has been it was done by Mark Konishi at Caltech early on in the 70s. Remains one of the key systems for our understanding of how sound is transmitted into interpretation. But that's done right here. So frequency patterns are analyzed for their content. Okay, I'll keep going. That information has to be passed to Broca's area, right? And this region is called the arcuate fasciculus, right? And Broca's area has to it says, oh, the word was thank you, or ngoy, right? And I say the word, for me to say thank you, I know I need to do certain things with my tongue, my larynx, the position of my lips. All of that has to be timed perfectly. Okay? That is brought up into the motor cortex. There's a premotor program and sent out. And as I produce the sound, I am continually modifying it through sensory feedback so that I know what my lips are doing. And, so I'm sort of exaggerating, but that is a wildly complicated engineering problem, and it's done like that. So if I were to build something to do this, to actually interpret sounds, repeat them, just that, that's a hard problem. I haven't told you to translate. Think about that one. So you guys, how many of you, I think all of you are bilingual, so you're not translating as I speak. Is that correct? Is that fair? Can somebody answer? I don't know. I'm not bilingual, I'm 1.5 lingual. My 0.5 is German, and when I speak German, I can do it, but my back of my head is translating and checking. Okay, so it's not quite just fluent. It's like, is that right? And so I think that's probably what you're doing, right? Okay, great. Let's do one more, and that is repeating a written word. Fundamentally different. I have a word here. Can everybody say this right now? Great, you did it. Just like that. Can any of you build a machine to do what you just did? Maybe. And again, I went all the way. I didn't just say recognize it. That's a character recognition. There's a lot of people working on this. But I have to go from the text to the speech, right? And by the way, Wernicke, Geschwind, model, okay? Tell me, what does this mean? You all know right away. What does model mean? Is it a woman in a dress at a fashion show? Right? Is it a type of car? Or is it conceptual? It's a conceptual model, but you instantly knew that difference. See, you had to do a whole bunch of processing, okay? So let's watch out. Your visual cortex analyzes the image, okay? That's right here, primary visual cortex. So there is the word model. It's got these figures in it. You don't, at this stage in your life, you're not doing what you did as a child. You're doing it instantly. So that circuit's already pretty hardwired in there, right? That information has to go to the angular gyrus, right? which is also where your word association goes from your ear. This one says, okay, now I'm going to go back through, and in this case, I go through, again, the arcuate vesiculus to Broca's area. So here, my written word came in like this, my spoken word came in like that. For whatever reason, if I say the word model or you read the word model, those magically get to the part of the brain that makes sense of the word model, whether it's spoken or read. So there's a region of your brain, as you'll see, that is about model. And as I think I'm going to show you, I can't remember all my slides, there's a region of your brain for tools that's separated from other structures, conceptual structures. Okay? So that again goes back here to Broca's area, and that program is launched again. I think the experiment's coming up. Okay. This is going to be interesting. Some people who cannot hear at all learn to read lips. That is, there's something about how your face moves that you can interpret that word. I'm going to tell you right now, I think this is going to work, but I'm going to try it in here, that you are always reading the lips. So if I say the word ba, the b, the lip closure and the open mouth afterward is a construct that your brain makes sense of. So here I'm going ba, ba, and you have no problem, right? Now I'm going to say a different word. I'm going to say the word da, da. You can hear the difference, right? Everybody close your eyes. Ba, ba. What did I just say? Okay, now close your eyes. Da, da. Great, exactly. What would happen if your eyes see one thing and your ears hear a different thing? Please watch this movie, okay? I hope this works. Oh, stop. What was he saying? Da, da. Is that what you heard? Okay, let's do this again. I'm going to play the same movie. I swear it's the same movie. I want you to close your eyes. Okay? Close your eyes. Open your eyes. Close your eyes. When your eyes are closed, he's saying ba ba. When your eyes are open, is this working for any of you? Okay. So it gets a little irritating. So let's think this through. That was an auditory visual illusion. Your brain looked at the mouth, and whether you're from Taiwan or Hong Kong or Beijing, B sounds are, or US, the B requires that your lips come together, and that D sound requires that your tongue and your teeth meet. I have no choice. There are other sounds that are harder, like R, because you can't really see what the tongue is doing. But the lips and the tongue is the giveaway. And your brain said, I don't even need to, I can just look at your mouth and say, know what you're saying. And turns out you could be wrong, which I really like. And it turns out also that your visual system overrides your auditory system. That if you see something that looks like they're saying da, even though the soundtrack is ba, by golly, my eyes never lie, right? And it turns out they did. So I like that experiment. An analogous one was done with a different sensory modality. You can't do this with a video. But it's a person who had a monitor very close to them. You'll like this one. and they had a little puff of air that would come off the monitor. And the person was saying DD, right? But with their mouth, but they got a puff of air on the cheek of the person hearing it, and they heard TT, right? Because it generates the puff, right, of air. And so your face gave you an auditory signal that slightly overrode your visual or your tactile or whatever. Great. I don't think I have much more for this morning. I did want to say that regions of the brain can be specialized, and we only found this out through current imaging techniques, to particular things, right? So tools, animals, and people literally occupy different parts of the brain, and you can, and they're fairly consistent from individual to individual. And stroke studies support this claim. And there are several different regions of the temporal lobe that there was an interesting individual who had a stroke that was messing up tools and other things. And he said the man started the car engine and stepped on the pancake. Well, it's a flat thing, okay? It's the pedal, but he stepped on the pancake because it's a flat thing, and that was good enough. And the person who said that had no problem, and they didn't think it was at all odd. It takes longer to process. So if you have these particular regions of the brain, here they are, right down at the base, right? So this is the association areas. You know, you hear here, and then these are processed up into there. You tend to have your friends here, your pets here, and your tools over here. And again, it's just fascinating how our brains have regionalized so much stuff, okay? Let's see, I just wanted to, oh yeah, let me just go a little further. When we talk about brains, we tend to think about our own, and I have to keep coming back to the fact that many of you will not necessarily do research on a human brain. You can still do tons and tons of valuable work on any brain. So I just wanted to give you a little sense about the history of the brain, not as a research study, but as something that has evolved, okay? So let me just get you a little more oriented. Everything we've been talking about is right up in here. This is a highly, highly, highly specialized area, and I'm just going to drill down a little more into this. So in a human, you can see that this extremely elaborated brain. This is frontal. This is the back of your head. There is the cerebellum, right? In a monkey, right away you can see that the sulci and the gyri are not as pronounced as in human. It's still, you have to think of the brain as a sheet, right? It's still a sheet-like structure, but it isn't quite so problematic to fill up that volume, so it's not quite so convoluted. And in fact, the interior of the brain are these large fluid-filled areas, right? In a cat brain, it's even less so. And look at the cerebellum, the primitive part. There's the cortex, right? The monkey cerebellum is back in here. Here is a rat brain. That's the cerebellum, right? That's the primitive part. There's the medulla oblongata off to the spinal column. There's essentially no sulci. Here's the frog. There's the front of the brain, right? Here's the cerebellum. This, by the way, is all olfactory, all smell. Vision is right here. Auditory is right there. This is all motor, right? So they're extremely, you see through evolution, ever and ever more volume going into the cortex, going into the things that we're doing, the language processing, the Wernicke center, Broca center, the things that we are very, very good at. The cerebellum, it was pretty much a done deed early on in evolution that we could control our movement, move around beautifully and wonderfully and excellently. We can get visual information and control motion. We could do that the minute there was movement. We have pretty good neural systems to run that. So what we're seeing in evolution is this elaboration of the communication parts of the brain, the parts that make us very social, interactive creatures, capable of standing up and talking for 2 hours. great. Here again, I want to drill down a little more into the cerebellum. Here are the sulci, right, coming in. These are the gyri, right? I should have pointed out one more thing here. Sorry. This is the brainstem, medulla oblongata. There's the complex motion and motor patterns. This region here is really fascinating. It's the diencephalon. And now this is a funny and rather poorly explored part of the brain. It's very primitive. But this is the part of the brain where it connects all of your sensory world, all of how you interpret the world, to the physiology of your body, your states of stress, your states of anxiety, your states of mood. This is the part that connects sensory world to hormone world. So when you're stressed, your cortisol levels are up. When you're frightened or when you're relaxed or in love or something, this is the part that adjusts all of that sensory information and prepares the entire rest of your body physiologically. So it's the connection between fast processing and sensing and these long-term states of your body, okay? It's got several capillary beds in it that deliberately connect neural signals into your bloodstream, okay? It's an absolutely wonderful region of the brain, very primitive. Here are the cortex, and this is what I want to do. So this is the sheet of paper, okay, that I lined up. And it's organized into layers from the outer layer deep down inside. And I want to give you an idea of the layering here. So these are, in this case, this is a 5-layered cortex. Layer 1, 2, 3, 4, 5. There's a 6th layer here. Some are 5. Visual cortex is 5, okay? Different regions of the brain have different numbers of layers. If I blow this up some more, this is giving you this idea of the connectivity. This is sensory, motor, memory. I can't read it. But what I want you to see is these regions of the brain are connected columnarly with splades of dendrites coming out like this. And these are connected horizontally through the brain. So you have connectivity this way and connectivity that way, okay? And it's extensive. If I look at just part of the, this has happened to be cerebellar, but any cortical group of neurons. These have large tracts going this way and huge connectivity happening in this direction, right? That's an amazing set of neurons there. And again, extreme connectivity. This is the trick of our brains. Importantly, as you use different parts of your brain, you either promote connectivity or sustain it. And as they are not used, right, they tend to not be sustained. It's a very plastic structure. Also importantly, that connectivity changes throughout your lifetime. When you're newly born, you have extreme connectivity of the brain, orofacial region. So you have reflexes that you have as a child, as a newborn or a young infant, that you no longer have. There's a whole series of these. And they're pruned out because they're no longer needed. So a classic reflex is if you do this motion on a newborn, their whole face will shift to the right, like that. And it's called the rooting reflex to feed on the mother, right? So you do this and you can drive a mother crazy by doing this to their child, so be nice, because they'll then want to feed. But that's a reflex that you don't have. You can do this all you want, but your lips aren't going to go flying off to one side, okay? The reflexes of how your hands and feet respond to falling switch from an adult protective to an infant pullback, right? Those are the two different ones. So a whole bunch goes on. The number of connections in your brain is peaked at about your current age. You probably have the most neurons that you're going to have in your life for the next few years. Sorry to my more mature colleagues, but they go away and they don't come back, So neuronal growth continues through adulthood, but neuronal death begins to override neuronal growth. Okay, these are grim truths. I think there's a gallery of neurons here just to give you a sense of the geometries of things we're going to deal with. And what I'd like to do is say from here forward, I'm going to drill down into the signal processing done by single units as opposed to regions of the brain, okay? And deal with, before I try and build up a circuit again, I'd like to deal with the actual transduction, how you convert external information to signals, okay? So tomorrow morning, we're going to spend time on the basics of how a neuron actually responds, okay? And I'm actually going to do a couple of different things, but hopefully we'll be able to do an experiment tomorrow afternoon. I may need a little assistance if I can. I've just got to build something between now and tomorrow, but it's not a big deal. And it's going to see if we can map receptive fields. So we're going to talk about a neuron, a single unit, how it transduces information. And then remember I told you that your mapping goes to specific regions? We're going to be able to actually measure a single unit's region, a single axon's region, by cleverly poking at each other, okay? So I think that's where I'm ending, but let me see. Yes, this is where we're going to begin tomorrow, but let me just ask, how many of you are familiar with this arrangement? Roughly half. So I will have to go over this, okay? So what we're going to do is I'm going to explain how these actually are able to respond as cells, what machinery you need, how signals can propagate, and what diseases affect those and what drugs affect those, okay? And the list is much larger than I could go into, so I'll just pick examples. So what we'll do is talk about the acquisition of information, its synthesis, transmission, and then communication. Those are the key functions of a neuron. For clarity, do not mix the two words up. The nerve is not a neuron. So for clarity, a neuron is one cell. A nerve is a group of cells. A ganglion is many nerves who have come into a single unit, and large ganglia are called brains. And also for clarity, we're going to leave brains and focus on single units and come back to brains eventually. But as I said, it was just this morning's lecture, a human brain, a mouse brain, a frog brain, a moth brain, a cockroach brain, they're all hard. But they're all structured fairly similarly in terms of extreme connectivity. Okay? That's the common theme. So I think rather than going all the way to 4, I think I'll just end here. I have a movie that I think I'll show tomorrow about the basics of how signal transduction happens in a neuron. It's a great movie, but I think I'll do that tomorrow. So without further ado, I think we'll adjourn for today. I might want one volunteer to help me get something before tomorrow afternoon. So if I can have one student volunteer, I'd appreciate it. What?
Do you need any specific background to?
No, I need toothpicks and little rulers. That's all. I need, like, let's see, how many students are here? We're going to work in teams of 2, so there's about 20. I need 10 little rulers and a box of toothpicks. That's the only thing I need. And if I knew where they were, I would go get them, but I don't know where they are. I know there's stores here. And they're on me. It's my treat. And what we're going to do is map receptive fields on you. It does take toothpicks and rulers to do this.
I can help you to volunteer.
Oh, you just volunteered somebody. Do you know what I mean by little rulers and toothpicks? What?
How long?
Oh, just something to measure centimeters. So what we're going to do, it's called receptive field mapping. And I do this in my introductory biology and introductory neuroscience class, is I want to know among you, can you tell how far apart 2 points are? And there's a point at which you can't tell whether it's 1 or 2. And I want to know how that varies over your body, right? And why it varies. And I'm going to leave you with an open question is, if you were to take 2 toothpicks separated by a known distance, this is the trick, right? And push them on, there's a point at which you can't feel whether it's 1 or 2. Why is that? And if you rotate them orthogonally, can you tell the difference? And if you can, how could that possibly be true? So we're going to check this hypothesis that your body is not just regionally spatialized, it's anisotropic. Okay? It's a cool experiment. Ultimately, why you'd like to know this, let me come back to neuroengineering, why we're going to do this experiment, is there's a new challenge in prosthetics, in devices that help you walk or reach, and that's getting sensory feedback. And that sensory feedback is oftentimes vibrational. It's called vibrotactile feedback. And they're using multiple vibrational stimuli to give you rich information. How should you space things? How should you move things? And this is a hard problem again. So again, I'm just going to keep you guys moving as something interactive to do every day. Today, the only thing you did was the ba-ba-da-da thing. It works. Did it work? Oh, good. Oh, good. It doesn't work on everyone. Some people ignore what they see. And they just hear and the correct soundtrack. Great. So that's it for today. Thank you. You guys are a fantastic group. So I look forward to abusing you for 4 more days. No clapping. No clapping. You've got 4 more days. Save it. Okay.